Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Spoilage of sugar and sugar products. Table of contents. Introduction to spoilage of sugar and sugar products. Sucrose, sugar syrup, maple syrup, honey. Introduction to spoilage of sugar and sugar products. Sugar is widely used in the food industry, both as a food product and as a food preservative. Sucrose is the most commonly used sugar and is readily available in nature. However, other sugars such as dextrose, lactose, and fructose are also utilized in the food industry. In recent times, polyols like sorbitol, mannitol, xylitol, and maltitol have been introduced as alternatives to sugar in sugar-free products. Common sugar-based products used extensively in the food industry include refined sugars, sugar syrups, maple syrup, and honey. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Sucrose. Sugar, sucrose, is obtained primarily from two major sources: sugarcane, Saccharum officinarum, and sugar beet, Beta vulgaris. Smaller quantities of sucrose are also produced from sugar palms 1%, sweet sorghum 0.05%, and maple trees 0.01%. Sugarcane and sugar beet undergo multiple processing steps to produce 99.9% pure sucrose. These processes include crushing and extraction, clarification, evaporation, crystallization, centrifugation, and filtration. Contamination source of sucrose: soil, raw materials, including sugarcane and sugar beet, handlers involved in processing, equipment and machinery used in production, poor hygienic handling practices, inadequate cleaning of sugarcane press knives and contact surfaces, airborne contamination. Spoilage of sucrose. The microbial population undergoes changes during the processing of sugarcane and sugar beet into sucrose. Spoilage causing microorganisms that contaminate raw juice depend on the specific microorganisms present during sugarcane and sugar beet processing. Common microorganisms found in sugarcane include Pseudomonas, Bacillus, Enterobacteriaceae, Lactobacillus, Erwinia, Leuconostoc, Flavobacterium, Xanthomonas, Corynebacterium, yeasts, and molds. Common microorganisms found in sugar beets include Pseudomonas, Arthropacter, Erwinia, Streptomyces, Bacillus, Clostridium, Flavobacterium, and yeasts. Yeasts linked to raw juice spoilage include Saccharomyces, Candida, and Pichia. Various microorganisms are associated with different major steps in sugarcane and sugar beet processing. In sugarcane, during the pre-harvest stage, microorganisms such as Flavobacterium, Lactobacillus, Xanthomonas, Enterobacter, Klebsiella pneumoniae, Pseudomonas, Erwinia, Leuconostoc, Bacillus, Corynebacterium, yeasts, and molds are present, leading to souring. In the post-harvest stage, Leuconostoc causes souring, sugar loss, and dextran formation. During crushing and extraction, Leuconostoc causes souring, Enterobacter causes sugar loss, and yeasts cause alcohol production. The clarification step shows no microbial contamination. In evaporation, crystallization, and centrifugation, thermophilic spore formers may be present. In refining, thermophilic spore formers are also found. In sugar beet, during pre-harvest, microorganisms such as Pseudomonas, Bacillus, Arthrobacter, Erwinia, Flavobacterium, Streptomyces, yeasts, and Clostridium are involved, causing acid production. In post-harvest storage, Bacillus causes slime formation, and Leuconostoc produces dextrins and leavins. During fluming and washing, Pseudomonas causes acid production, while Flavobacterium contributes to corrosion. In extraction, Bacillus sterothermophilus leads to acid production. Clostridium and thermophilic cocci cause sugar loss, and thermophilic cocci also produce hydrogen sulfide. During crystallization, thermophilic spore formers introduce spores into the final product. Bacteria linked to raw sugar spoilage include Bacillus sterothermophilus, Bacillus coagulans, Clostridium thermosaccharolyticum, Desulphotomaculum nigrificans, and various mesophilic bacteria. Yeasts present in raw sugar are predominantly osmophilic, with common genera including Xerosaccharomyces, Hansenella, Pichia, Torolopsis, Candida, Decoromyces, and Endomycopsis. Molds in raw sugar are typically xerophilic molds. Sugar refining processes destroy microorganisms when present. However, recontamination can occur from surviving thermophilic spores. Microorganisms associated with raw and refined sugar can cause various defects in the final product. In raw sugar, Zygosaccharomyces roxii can cause sugar loss. Xerophilic molds can lead to the production of invert sugars, acid formation, and an increase in water activity. In refined sugars, surviving thermophilic spores may introduce spores into the final product. Preservation of sucrose. Sugar has a naturally low water activity, making it microbiologically stable and suitable for storage at room temperature for several months. Spoilage occurs when sugar absorbs moisture. Therefore, during storage, vermin should be excluded to keep the sugar dry. Sugarcane and sugar beet should be thoroughly cleaned and stored in a controlled atmosphere. Fungal growth can be inhibited by using gases such as 6% carbon dioxide and 5% oxygen. Chemical preservatives are applied during the sugar refining process. Hygienic conditions must be maintained throughout the entire sugar refining process. Pasteurization is employed to remove microorganisms from sugarcane or sugar beet juices. Additional preservation methods for sugarcane or sugar beet juices include ultrasound, irradiation, high-pressure processing, and pulsed electric field treatment. Sugar syrup. Liquid sugar made from cane or beet sugar is produced by dissolving crystalline refined sugar in water. It typically contains a sugar concentration ranging from 66 to 76 degrees Brix. Sugar syrups are widely used in products including confectionery, beverages, bakery goods, ice cream, jams, jellies, and meat products. Contamination source of sugar syrup: water and sugar used in production, equipment and machinery involved in processing, poor hygienic handling practices, contaminated raw materials, airborne contamination, improper storage conditions. Spoilage of sugar syrup. Adding water to refined sugar to produce liquid sugar increases its susceptibility to microbial growth. Sugar syrups can be contaminated by various microorganisms, particularly spore-forming bacteria, yeasts, and molds. Osmophilic yeasts and xerophilic molds are the primary spoilage organisms in sugar syrup. Common spoilage yeasts include Zygosaccharomyces, Ruxi, Saccharomyces cerevisiae, and Saccharomyces meles. Common spoilage molds include Aspergillus glaucus and Penicillium expansum. These microorganisms can produce foul flavors through the formation of alcohol, lactic acid, and other organic acids. Preservation of sugar syrup. Sugar syrup can be preserved by filtration, heating, or ultraviolet light treatment to remove or inactivate microbial contaminants. Pasteurization can be applied at 65 degrees Celsius for 30 minutes, 75 degrees Celsius for 25 seconds, or 85 degrees Celsius for 15 seconds to eliminate microorganisms. Pasteurized sugar syrup can be stored under refrigerated conditions to extend shelf life and prevent microbial contamination. Additional preservation techniques include ultrasound, irradiation, pulsed light, high-pressure processing, and pulsed electric field treatment. Maple syrup. Maple syrup is derived from the sugar maple tree, Acer saccharum. Maple sap consists of 95 to 99% water, along with minerals such as calcium and potassium, 2 to 5% sucrose, various organic acids, and vitamins. The sap is collected by drilling and tapping the trees. Contamination source of maple syrup. Uncharacteristic environmental changes, improper sap storage conditions, unsanitary processing practices, equipment used for sap collection. Spoilage of maple syrup. Sap is sterile while inside the tree's xylem, but once exposed to the environment, it becomes highly susceptible to microbial growth. Maple syrup's water activity, ranging from 0.83 to 0.86, prevents the growth of bacterial pathogens. The most common bacterium found in sap is Pseudomonas fluorescens, along with certain yeast species. Spoilage-causing bacteria include Pseudomonas aerobacter, leuconostoc, Enterobacteriaceae, and Bacillus species. Spoilage-causing fungi include Phyllophora, Ascocarine, Phoma, Alternaria, Penicillium, Fusarium, Gliocladium, Cephalosporium, Cladosporium, Candida, Trichoderma, and Actinomycetes. Different microorganisms are associated with maple syrup at various stages of processing, leading to specific defects. During the collection and tapping stage of maple sap, bacteria such as Achromobacter species and leuconostoc species can cause reduced sap yield and ropey sap. Bacillus species and Flavobacterium species may lead to reduced sap yield, ropey sap, and buddy sap. Chrysobacterium species, including Epilithonomonas, and members of the Enterobacteriaceae family, can cause reduced sap yield. Klebsiella species may cause ropey sap, while Micrococcus species and Pseudomonas species can lead to reduced sap yield and buddy sap. Plantibacter species and Staphylococcus species are associated with buddy sap. Fungi in the collection and tapping stage include molds such as Acremonium species, including Cephalosporium species, Alternaria species, and Ascocarine species, which can reduce sap yield or quality. Aspergillus species, including Eurotium, Cladosporium, Fusarium, and Penicillium species, can reduce sap yield or quality and cause surface mold. Yeasts like Candida, Cryptococcus, Rhodotorula, and Saccharomyces species can reduce sap yield or quality and lower syrup grade. During storage, bacteria such as Aeromonas species, Brevibacillus species, Brevundimonas species, and Cloivera species can contaminate the sap. Fungi like Cytospora species, Mucor species, and Rhizopus species can cause surface mold. In the packaging stage, bacterial contaminants may include Agrobacterium species and Bacteroides species. Some spoilage defects in SAEP include green sap, caused by fluorescent pseudomonads, red sap, caused by red pigmented yeasts and certain bacteria, milky sap caused by bacillus species. Preservation of maple syrup. Store collected sap at 40 degrees Celsius until further use to control microbial growth during processing. Clean all equipment regularly to prevent contamination. Use filtration, evaporation, or ultraviolet treatment to remove or inactivate microbial contaminants in syrup. Apply pasteurization to sugar syrup, 65 degrees Celsius for 30 minutes, 75 degrees Celsius for 25 seconds, 85 degrees Celsius for 15 seconds. Store pasteurized sugar syrup under refrigerated conditions, or can it to extend shelf life and prevent microbial contamination. Consider advanced preservation techniques such as ultrasound, irradiation, pulsed light, high-pressure processing, pulsed electric field. Honey. Honey is a natural sweet substance produced by honeybees, defined as the nectar and saccharine exudates of plants, gathered, modified, and stored in the honeycomb by Apis mellifera. It is widely used in the food industry in products such as condiments, salad dressings, barbecue sauce, peanut butter, dairy products, meats, beverages, snacks, bread, cereal products, and candy. Composition typically includes 15 to 21% water, 30 to 35% glucose, 35 to 45% fructose, 1 to 3% sucrose, 10 to 12% maltose, along with organic acids, minerals, proteins, amino acids, and enzymes. Water activity ranges between 0.54 and 0.75, with an acidic pH between 3.2 and 4.5. Contamination source of honey, flowers, and pollen. Digestive tracts of honeybees, dirt, dust, and air. Food handlers, equipment used. Spoilage of honey. Microorganisms capable of surviving in honey are those that can tolerate its high sugar concentration, acidity, and other antimicrobial properties. Honey can be contaminated by a variety of bacteria, yeasts, and molds. Most bacteria and microbes remain dormant in honey because of its antibacterial nature, which prevents their growth and reproduction. Yeasts and molds can survive in honey, and certain vegetative and spore-forming bacteria can also persist. Osmophilic yeasts are the primary cause of honey spoilage, as they are more capable of growing in honey compared to bacteria and molds. Bacterial species detected in honey include Bacteridium, Bacterium bacillus, B. cereus, and B. pumilus, Brevibacterium, Enterobacter, Flavobacterium, Micrococcus, Neisseria, Pseudomonas, and Xanthomonas. Yeast species found in honey include Rhodotorula, Dibariomyces, Hansenula, Lipomyces, Ospiridium, Pichia, Torulopsis, Trichospora, Nematospora, Schizosaccharomyces, Schwanniomyces, Torula, and Zygosaccharomyces. Mold species associated with honey include Ascosphaera, Aspergillus, Cephalosporium, and Penicillium. These microorganisms can produce alcohol, carbon dioxide, lactic acid, and other organic acids, leading to undesirable flavors in honey. Preservation of honey. Honey naturally acts as a preservative due to its strong antimicrobial activity, which is attributed to its osmotic effect, acidity, hydrogen peroxide content, and phytochemical components. It exhibits bactericidal effects against many pathogenic microorganisms, including Salmonella species, Shigella species, Escherichia coli, Vibrio cholerae, and various gram-negative and gram-positive bacteria. Pasteurization is carried out at 71 degrees Celsius for 5 minutes, followed by rapid cooling to 38 degrees Celsius. After pasteurization, honey can be stored under refrigeration or canned to extend its shelf life and prevent microbial contamination. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!